Hi, everyone. Welcome to What's New in MPMI. While we're waiting for people to log on, I see the room is starting to fill up. Please type into the chat where you're from. It's always fun. We have such an international community. It's always great to see where people are coming from. So why don't you type that in and we'll be able to see. I have Adrienne Hamanjian from France, Charlotte Heitmüller from Germany. Oops, it's going very fast. Diego Gutierrez Castillo from Peru, Bilal from Kashmir, JJ in Norwich, Satish from UAPB, Karun Wilson from India, Kire Heleth Munoz Jaimes from Mexico, sorry about that, Valdir Junio Vaz Moreira from Brazil, we have Maria Eugenia from Argentina, Vic Nicolas from South Africa, Henrik Stott, hi Henrik, nice to see you. Melanie Mendel, from, Henrik and I were postdocs together. Melanie Mendel from Utrecht, Mojito Hussein from Perth, Australia, Yvonne Mukuka from Australia, Raymond Van Dellen from the Netherlands. I'm going to have to stop, but I think you can see we're a very international community and we have people joining from all over, which is exactly the goal of this event. So I'm really excited. So welcome to What's New in MPMI. I'm Jean Harris. I'm the editor-in-chief of the MPMI Journal. And with me today is Catherine Jones from Florida State University, who is one of our senior editors and is going to be co-host today. And our speaker today is Kyungyong Seong, who is the winner of our Graduate Student Paper Award for MPMI this past year. He is a graduate student with Ksenia Krasileva. And I want to point out that one of the goals of our What's New in MPMI series is to increase access to our science across the world. It's free, just like the journal is open access. So you have access both to the papers and to the talks. And really it's to promote connections. One that's been the silver lining of the pandemic is to realize there are lots of different ways we can connect. And this is one of them. And it's been a real pleasure for me to meet people virtually around the world like this. It also gives people a chance to connect with our authors more directly, not just on the pages of the journal, but here in person, and to be able to hear them talk in their own words about their work. And finally, it's also to promote the career of young scientists. And so I'm especially pleased that today we are featuring Kyungyong Seong, who is our Graduate Student Award winner. And I want you to mark your calendars for January 17th, which is our next seminar. We won't be having one in December. Next seminar will be featuring Emma Gachimo, who will be talking about a very interesting interaction between Brady rhizobium, which most of you know as a legume symbiont, and its interactions with Arabidopsis and the way it alters its root architecture via manipulating auxin transport. So that registration is going to be coming up soon, and I recommend that you register for that in advance. And finally, don't forget, we're actually going to be having an in-person Congress in 2023 in Rhode Island in July. Keep your eyes out for that on the IS-MPMI website. So today I have the pleasure of introducing Kyungyong Seong, who is, as I said, our Graduate Student Award winner. His paper attracted my eye instantly when it first appeared, actually, even before it got into first look, when I was watching it go through the system. Such an interesting computational approach to really opening up a new way for us to look at effectors and do this genomic structural modeling. And it's even more fun for me I'll give you just one little taste of this before I turn it over to him, is that he actually did this as a rotation project during lockdown. So I think that speaks very much to the creativity of his advisor, Ksenia, and also shows that a very useful way of using this time. And he gained a lot of wonderful skills and entered into this whole new area that he's really opening up for the whole community as well, not just for himself. And that's why this was something I especially wanted to share with you. Okay, Kyungyong, I'm going to turn it over to you. Oh, and I just want to say, pardon me. At any point during this seminar, you may type your question into the Q&A box at the bottom. We will be turning off the chat, and it will just be going to Q&A, and then Catherine Jones and I will take turns reading the questions at the end. Look forward to hearing from you, and Kyungyong, you can share your screen.
Okay. All right. Thank you for the introduction. It's good to see you all, everyone. Although I don't really see anyone on my screen, I can feel your presence. So thank you for having me and joining me for today's talk. The title of the talk today is supposed to be Computational Structural Genomics Unravels, Common Folds and Novel Families in the Secretome of Fungal Phytopathogen, Magnaporthe oryzae. But given what I'm going to talk about, I decided to give a little more generic title, Elucidating Effector Evolution Through Predicted Structures. So the world population is growing rapidly. Although the amount of food required to feed them will be increasing exponentially, the amount of food we can produce will be increasing only linearly. And we might hit a time point when we can no longer feed a growing world population with the current agricultural systems. There are many difficulties against most effective or efficient agricultural system. Those could include climate changes, global warming, or limited water usages. But at the same time, pests have significant impact. According to the estimation by FAO, about 20 to 40% of the food production are lost to pathogens every year. And among pathogens, I will focus on fungal pathogens in particular. Here I'm highlighting some of the important fungal pathogens, including rust pathogens, gray mold, Fusarium wilt, Plumeria graminis or powdery mildews, blastopathogen, and corn smut pathogens. So one of the important missions in the MPMI community is, of course, to understand how these fungal pathogens infect the plant species and how we can secure sufficient food for the current and future generations. Fungal pathogens secrete molecules called effectors. These effectors often target important host molecules to suppress plant immunity or modify host cellular activities or hijack metabolic pathways and use the plant host for pathogens benefits. In turn, plants also have evolved very sophisticated immune systems. In the inside of the cell, plants have intracellular immune receptors that can recognize the intracellular effectors. Or at the plant surface, there are cell surface immune receptors that can recognize apoplastic effectors or other types of conserved signature of pathogen molecules. The recognition of these molecules can lead to downstream immune responses that can lead to resistance against invading pathogens or healthy phenotypes of the plants. Although, so one big part of the MPMI community is, of course, to understand what these effectors are and how these effectors functions. And although these days it's much more common for us to collect the field isolates and sequence and annotate their genomes, still elucidating what effectors are is a tricky question. And this is because often you might end up with a long list of hypothetical or conserved expressed proteins without any types of functional annotations that can tell you what these effectors might be. In the meantime, the structural biology has elucidated important evolutionary aspect of effector evolution. It is that although these effectors may be not related by their primary sequences, these effectors may resemble in terms of structures. And these sequence-unrelated, structurally similar effectors can be called sus-effectors in general. Here, we're looking at one of the very well-studied sus-effector class, which is a Mox effector from Magnaporthe oryzae, and Lars effectors from Leptospirea, and Ralph effectors or RNases effectors from powdery mildews. Also, fold effectors from Fusarium oxysphorum. The repeated occurrence of such vectors signify their importance for pathogen evolution and pathogens survival. In the meantime, there has been increasing amount of study in molecular biology, structural biology, and also evolutionary biology that has been highlighting active interplay between plants' intracellular immune receptors and pathogens susceptible vectors. For instance, host pathogen may evolve intracellular immune receptor that can recognize this susceptible vector. And recognition of this susceptible vector can lead to resistance against invading pathogens. In turn, pathogens may lose susceptible vectors tactically in order to evade immune recognition and regain pathogenicity. And of course, pathogens may be able to re-pick up these vectors to gain some pathogenicity against different cultivar of the plants. In some cases, pathogens may accumulate point mutations on the surface of the proteins to again evade immune recognitions. And then the intracellular immune receptors may accumulate compensating mutations that can regain such recognitions in general. So the active interplay between these intracellular immune receptors and susceptible vectors signify importance in susceptible vectors, and then continue the war between susceptible vectors between the plant hosts and fungal pathogens. So identifying novel and existing classes of susceptible vectors will be an important part of vector biology. However, we cannot solely rely on structural biology to do the work. According to this review published in this year, for the last decade or so, we were only able to identify about 6 to 7 different susceptible vector classes, while there may be many more. So although the contributions of structural biology has been really great, we will need an alternative way to identify susceptible vectors from lots of pathogens that we're interested in. Structure prediction could be an alternative way. However, applying structure prediction was not necessarily easy either. Back in 2018 or 19, we were getting a lot of good structures out from template-based modeling. In this method, we started from query sequences, and then we identify homologous sequences from the query sequences that have already solved the structures. And by mapping or threading the query sequences into the existing templates, we will get something out of it. But remember, susceptible vectors are typically sequence unrelated. So in the very first step where we have to identify the sequence similarity between query and template, we might actually fail already. In another case, there was a template-free modeling, and fragment assembly might actually represent a good example of it. In fragment assembly, you start by sampling little tiny fragments of the protein structures from the solved existing protein database, such as PDB. And you assemble these little tiny fragments that is typically composed of the 6 to 12 amino acids to assemble something that is optimal in terms of energy and given primary sequences. Although template-free modeling had some successful examples, this method typically failed really quickly because the amount of space that you have to explore to spot the right reasonable protein structure is typically huge, and exploring entire space with the computational method was not typically feasible. So template-free modeling would break down pretty quickly as the size of the protein increases, or the complexity of the protein increases. In that sense, the debut of AlphaFold back in 2018 was actually very, very exciting because AlphaFold is a free template-free modeling algorithm. The difference of AlphaFold was, of course, the incorporation of deep learning-based algorithm that learned some sort of universal rules in translating primary sequences to 3D structure. And here I have this illustration of how AlphaFold might be actually folding one of the target sequences given in the competition. So if you compare the performance of AlphaFold that was reported during the critical assessment of structure prediction 13 that happened in 2018, it was much great even greater than other top scoring predictors back in 2006 and 2016. And of course, the performance of AlphaFold 2 was even greater, exciting the entire community. One of the important theme of AlphaFold was, of course, including some sort of coevolutionary information. Imagine you are looking at protein structure and we're now zooming into the core of this protein. There might be a pair of protein amino acids that are interacting with each other. And right here, okay, right here. And imagine there is now a mutation that happened in this red amino acid to green amino acid that might destabilize the protein structures. What could happen is a compensating mutation of this green residue to red residue. And this can be actually generalized. That is, the amino acids in proximity in 3D structure might co-evolve together to stabilize their interactions. So if we happen to collect a lot of homologous sequences and align them together, we may be able to capture such coevolutionary signature or the pairs of residues that are evolving together. And then we can translate this information back into the 3D structure. That is, we can position these pair of amino acids in close proximity in 3D structure so that when the protein gets folded, they are nearby. In this way, we can kind of limit the large, huge protein structural space that we have to explore, and then get to the right protein folds pretty easily and more accurately. Another important theme of deep learning-based algorithm was, of course, to learn some sort of universal rules in translating primary sequences to 3D structure information. And although we're not super interested in this, one active research area is a synthetic protein design. And this is a synthetic protein design or target generate human-made proteins so that these proteins can be folded into some desired protein structure. And what was exciting to us was that this deep learning-based algorithm was able to predict the folds of these synthetic proteins, even in the absence of coevolutionary information. This was great news to us because in many cases, effectors do not have a lot of homologous sequences from which we can extract the coevolutionary information. However, this suggested to us that deep learning-based algorithm may be able to capture the folds of effector even in the absence of coevolutionary information. So gaining some confidence on the fact that we can use deep learning-based protein structure predictors to predict the folds of effectors from fungal pathogens, we decided to apply this methodology into the prediction of secretome of fungal pathogen Magnaporthe oryzae. As a deep learning, the protein structure algorithm, we use the TR-zeta, which is represented by this paper on the top. And then we also included another very popular structure prediction tool called ITeser, which largely relies on homology-based modeling. Because we're living in 2022, you might ask, why didn't we use AlphaFold? And I want to note that the source code of AlphaFold 2 became available only in July 2021. And when we were starting our study back in 2020, TR-zeta was one of the best performing algorithms that we could access. So that now leads to our first paper where we apply TR-zeta and ITeser in the secretome of Magnaporthe oryzae and reveal a lot of interesting features about this pathogen. The very first thing that we wanted to confirm was that TR-zeta can predict the folds of effectors in the absence of homologous structures. So we collected a lot of proteins with the solved structures from the Magnaporthe species, and then let ITeser and TR-zeta predict their structures. The red dots indicate the Magnaporthe proteins that are not secreted, and then the blue dots indicate the secreted proteins from Magnaporthe oryzae with the solved structures. And the the x-axis and y-axis indicate actual precision that is acquired by comparing the predicted structure from either ITESR or TR-zeta to the actual solved structure available in the PDB. This ranges from 0 to 1. Typically, when the TM score or the actual precision is higher than 0.5, we say the fold of a vector, fold of a protein is roughly or correctly predicted. And what we saw was that TR-zeta was able to predict the fold of MOXIF vectors, especially AVR-PP, AVR-PIT-T, AVR-PIC-D. Although ITESR was able to predict AVR-PI and MOXIF vector structures, that was because ITESR picked up some of the homologous templates that showed some low sequence identity. And overall, what we gained the confidence that TR-zeta, even in the absence of homologous templates, would be able to predict the folds of vectors. At the same time, we examined whether TR-zeta could actually predict folds of other types of vectors. So we explored the PDB database in order to collect some of the secreted proteins from Ascomycota and Basidiomycota with the solved structures. And we let TR-zeta predict their structures. Again, we have actual precision on the y-axis. And what you see on the x-axis is estimated precision. It's a metric that TR-zeta outputs to tell us how accurate that prediction might be. What we noticed at the time was that TR-zeta was able to predict many of these vector structures, although there were some missing. And at the same time, there was a good linear correlation between the estimated precision and actual precision. That is, the protein structures that were predicted to be good will be actually good when we compare the predicted structure to actual precision, to actual structure. So that meant that we can rely on estimated precision to pick up reliably predicted folds versus unreliably predicted folds. So knowing that, we decided to apply ITESR and TR-zeta at a secretome-wide scale. We first started from the proteome of Magnaporthe oryzae, extracted all the secreted proteins based on the N-terminal signal peptides, and removed membrane-anchored or membrane-localized proteins, and then filtered out some more proteins based on the size or overlapping domains at the N-terminal region where the signal peptide was predicted. And we ended up with about 1850 proteins or so in our final list. And then we let ITESR and TR-zeta predict their folds. What we noticed in these distributions in the final output was that about 500 proteins that were not predicted by ITESR could be predicted by TR-zeta. And at the same time, about 600 proteins that were predicted by both ITESR and TR-zeta. Among those, about 500 proteins showed a greater estimated precision on the TR-zeta models. That indicated that deep learning-based algorithm was actually outperforming homology-based modeler in that it can predict many structures that could not be predicted by homology modeler in the absence of homologous templates. And at the same time, even when homologous templates are present, the deep learning-based TR-zeta was performing much better than homology-based modeler, ITESR. And although there were about 150 proteins that were only predicted by ITESR, these were not super significant. And that was because these include a lot of highly disordered proteins. Somehow ITESR picked up some unrelated templates from like humans or yeast that are also highly disordered, and then forced the mapping through these templates, giving us some sort of high estimated precision, which is unlikely true. So now we have some predicted structures. What can we do? We first visualized the structures of many violence and aviolence proteins that do not have any types of functional annotations. And comparing the structures of these violence vectors or aviolence vectors into the existing protein databases also gave us some sort of insights into what these proteins might be. For instance, in case of MOCDIP1 and MOCDIP5, we're not able to predict any types of PFAM domains at all. However, comparing the predicted structure of MOCDIP1 to the existing databases gave functional annotation for pectin lyases, and similarly for MOCDIP5, although it didn't have any types of PFAM annotations, it was predicted to be metallopeptidase. And also, even when some PFAM annotation was available, just like in case of MOCDIP10, we could annotate additional domains at the C-terminal region based on the structural similarity. In particular, here we have a beta sandwich fold, which typically is involved in recognition of some molecules or proteins. Another example might be this. These are two hypothetical proteins without any types of PFAM annotations, but based on structural similarity, we could annotate them as a DNases with some sort of N-terminal or C-terminal binding modules. So that indicated to us we can utilize the structure information to provide functional annotations for some hypothetical proteins without any types of sequence-based annotations. Well, knowing that predicted structures can provide more information about putative functions of hypothetical proteins or hypothetical effectors, we also wanted to examine whether we could identify existing classes of sus effectors that I explained before, especially Mox effectors. When we are doing sequence-based clustering, we typically rely on network analysis. Imagine these dots are sequences, and then when there are sequences similarity, we can connect them using these lines here. So for instance, in these cases, we will identify based on sequences similarity, 3 distinct subclusters that show that are homologous to each other. What we could do is, of course, overlaying structure information on top of this. We can compare structural similarity between 2 proteins, and if there are significant structural similarity, we can connect them with the lines. And what could happen is, of course, that all these singletons and sequence-related subclusters may be structurally similar and can be clustered into a larger network that may represent the sequence-unrelated structurally similar effector group. So we applied this technique to see if we can recover Mox effector clusters. Originally, based on sequence information alone, we could identify 3 different sequence-based subclusters and 2 singletons. And we used the predicted structures of TRZ and ITESR to compare their similarity and connect them. And what happened was, of course, that these sequence-related subcluster and singletons can be connected into a larger Mox effector clusters, indicating to us that we can use a structure-based similarity searches and clustering to indeed identify the existing classes of sus effectors. Because we were interested in sus effectors, we also examined our clustering outputs to see whether there are any novel sus effector classes. And indeed, we found quite a few. For instance, in this case, we had 3 sequence-related subcluster and 2 singletons, and they could be connected by structural similarity. And as you can see, the structures were quite similar to each other. And what was also interesting was that we were able to identify a structurally similar secretory protein from Leptospirea, which is another fungal pathogen that infects plant species. Similarly, this cluster had a core fold composed of 4 beta sheets and another 4 beta sheets forming beta sandwich fold. Again, there were 3 sequence-related subclusters and 3 singletons, and those were all connected by the structural similarity. And they were sharing all the common beta sandwich fold together. And again, as we just saw in the previous slide, we were also able to find structurally similar secretory protein from another fungal pathogen, Cladosporidium fulvum. And collectively, that indicated to us that some sus effectors may be common across many different fungal pathogen species. And so far, together, what I want to highlight here is that deep learning-based protein structure prediction tool works much better than homology-based modeler. And using predicted structures, we can annotate hypothetical or conserved proteins without any types of functional annotations. Using structure-based similarity searches and clustering, we can identify existing and novel classes of successive vectors. And then lastly, some of these successive vectors might be common across different fungal pathogens. And by the time we were wrapping up our project, the source code of AlphaFold 2 became available to us. So it was actually good timing for us to try AlphaFold and then predict the folds of secretory proteins across many different fungal pathogens, compare the similarity between them to see if we can identify any other novel classes of successive vectors or common classes of successive vectors that we just saw in Magnaporthe oryzae. So we applied AlphaFold on the same set of data sets from Magnaporthe oryzae. What we saw was that even AlphaFold was able to predict higher number of predicted secreted proteins structures, but also the accuracy reported by the AlphaFold was even greater than what we saw in TR-zeta or iTezer, indicating that the performance of AlphaFold was really, really enhanced a lot. So gaining confidence, we expanded our study at a competitive scale. And I want to highlight that our paper just got accepted, and you might be able to see the revised version of the paper in press very soon. So I'm really happy to share this. So we started by collecting different fungal pathogen species from Ascomycota and Basidiomycota. And these pathogen species included not only Magnaporthe oryzae, but also other important fungal pathogens like Fusarium oxysporum, or corn smut pathogen Eustylagomades, rust pathogen Puccinia graminis, gray mold Botrytis. We also included non-pathogenic saprotropic species per order and subdivision as a sort of controls. And we also collected Phytophthora infestant Secretome as an outgroup. And then we let AlphaFold to predict the entire secreted protein structures from all these different fungal species and compare their structural similarity and cluster them together to identify novel successive vector clusters. And we noticed that some fungal pathogens, especially a lot of biotropic pathogens like Bullomeria graminis, powdery mildew, or rust pathogen Puccinia graminis, and Eustylagomades had species-specific successive vector clusters. For instance, here, the cluster 5 included a lot of RNases-like vectors, and Bullomeria graminis had more than 400 RNases in these clusters. And similarly, cluster 27 included known violence vectors, AVR-SR35, and it was specific to Puccinia graminis. So we started to examine individual cluster in order to see some interesting evolutionary feature associated with each cluster. The very first thing that we found was that known violence or aviolence vectors actually represent new successive vector classes. Again, this is another network graph where the 3 sequence-related subclusters and 2 singletons were connected by structural similarity. AVR-SR35 belonged to these purple sequence-related subclusters, and we had some other structurally similar effectors that do not have any types of sequence similarity. That indicated to us that AVR-SR35 is another example of successive vector. And recently, the cryo-EM structures of AVR-SR35 and its cognate receptor SR35 has been solved. And that again indicated to us that there is active interplay between the successive vector class and planimin receptors. We were also able to examine some of the evolutionary features associated with the vectors. In case of cluster 25, that was nearly specific to Puccinia graminis or rust pathogen. These cluster members didn't have any types of functional annotations based on sequence information. However, structurally, they resembled hydrophobins. Indeed, when we examined the entire connectivity of the clusters, we found that cluster 25 had a similarity, structural similarity to cluster 52, in which the members in this cluster were all annotated as hydrophobins at the sequence level. And that indicated to us possibly this cluster 25 that forms a novel susceptor class could have originated from conserved secretory protein that are commonly found in fungal species, but rather the extreme divergence and radiation of these susceptors were very specific to pathogen, leading to pathogen-specific susceptor class. Luckily, there were some functionally characterized effectors in cluster 25, and then we noticed that the localization of these effectors were all different. Some of them were localized into chloroplast, some were going to nucleus, some were going to cytoplasm. And that supported that possibly the rapid divergence and radiation of such effectors might have brought functional diversification of these effectors as well. Another exciting example came from Eustylagomades or corn smut pathogen. According to the study conducted in 2014 by Breforth et al., when the authors deleted entire genomic segment that is shown here, the corn smut pathogen turned into epiphyte. That is, although the pathogen was able to still live inside the plant host, corn, it was no longer able to cause diseases. And then TIN2 at the time was able to be found as an important virulence vectors. These entire genomic clusters include about 20 secretory proteins, and they can be grouped into 4 different gene families, and then 5 different singletons based on sequence similarity. But what we found was that all these hypothetical proteins actually share structural similarity, indicating that they could have come from a common origin. What could have possibly happened is that the ancestral TIN2 fold underwent duplication event, and subsequent expansion of the clusters could have resulted in such genomic organization that we currently see. At the same time, Breforth et al. Reported that there were no pathologous sequences in other chromosomes. However, we also were able to identify structurally similar effectors that are translocated into chromosome 5 and chromosome 20, suggesting that elucidating entire picture of effector evolution requires a structure information. Another interesting aspect of TIN2 effector evolution was that its evolution was a little more drastic. We found 4 different clusters that have core TIN2 fold colored in blue, surrounded by long disordered stretches colored in orange. And as we compare the sequence length variations within the clusters and between the clusters, we also saw significant variations. And by the way, the annotations for these TIN2 disorder fusion proteins can be supported by transcriptomic data. So what we see here is actually real. And this suggested to us that core fold may undergo some sort of duplication and fusion events, which later may be extended or contracted for further diversification. And although I didn't really have enough time to introduce entire evolutionary feature of effectors, our paper overall highlights divergent evolution driving effector evolution. More specifically, the ancestral protein may undergo duplication events, and one of the pathologs may diversify or diverge, occupying different evolutionary space. And then repeated occurrence of duplications and divergence may lead to the formation of sequence-related subclusters in different regions of this evolutionary space. If you're only relying on sequence information, you might be able to identify 4 different sequence-related subclusters that do not share any types of other similarity, But by overlaying structure information, you can fully rebuild the entire connectivity of these clusters and deeper evolutionary perspective on the effector evolution. That's what our final figure highlights, where effector evolution is driven by divergent evolution. And I want to highlight that not all effectors might be evolving for novel functions. That is, some of the effectors might be genetic noises. Sometimes the purifying selection can be relaxed, and the effectors might be accumulating rapid mutations that was not accessible before. Sometimes effectors might be undergoing extensive drift without having definitive roles and functions. And these effectors might gain useful functions, useful virulence functions in the long run, but others might be lost during the evolution. One challenging part of divergent evolution is that, of course, the functional diversification might be followed. So even though we might be able to identify novel and existing sus-effectors using structure information, we may not be able to assign a single definitive role in each effector class. That means the molecular biology work guided by evolutionary biology will be still essential in our understanding of effector evolution. Before wrapping up, I just want to highlight some new opportunities in computational structural genomics, given how fast this field is moving forward. Based on our study, there were about 600 secreted proteins, the structures of which could not be predicted by AlphaFold 2 and TRZeta. And we think that the trend of these data sets will be very similar to what we suggested in our MPMI paper. That is, many proteins will be still very small, less than 300 amino acids. And although some of the proteins may not be foldable because they are highly disordered, others will have rigid foldable structures, and AlphaFold just failed to predict them. In that cases, you might consider incorporating other structure prediction tools. Especially these days, we have access to language model-based protein structure predictors like OmegaFold, IS-MPMI, RGN2. And also recently, the AccuFold that relies on coarse-grained representation became available to us. So applying these different techniques might be able to rescue some of the secreted protein structures that were not predicted by AlphaFold, deepening our knowledge on effector evolutions or effector features. At the same time, you might consider expanding the competitive study. There are already 200 million protein structures available in the AlphaFold protein database. And recently, IS-MPMI released their own database that hosts about 600 million protein structures. By including existing protein structures and predicting structures for your favorite pathosystem, you may expand the competitive study to include nematodes, insects, bacteria, or mycetes. Or at the same time, you might consider comparing your fungal species against other fungal species that infect non-plant species like humans and mammals. Such larger competitive study may deepen our understanding on how plant pathogens evolve, and that may bring us new insights into plant pathogen interactions in general. There are already many interesting stories out there that I want to highlight. These studies already incorporate AlphaFold 2 to understand the composition and regulation, evolution of fungal pathogen secretome. So it will be exciting to see the same methodology applied to your favorite pathosystem and your favorite MPMI questions. With that, I want to acknowledge all the members in Cross-Level Lab, especially Ksenia has been amazing PI and mentor and supporter in many different aspects. And also I needed to thank Berkeley and PNB for their generous support. And again, the MPMI community and the journal for giving me such a great opportunity for me to talk about my science and get to the broader audience. And also my big supporter, Toby and Apollo, I think you saw Toby sitting right here. Although they were not co-authors of the paper, they have been providing great mental care for me for the last 2 years or so. So with that, I'd like to thank you for joining and listening. And I'm happy to take any types of questions.
Wonderful. Thank you so much, Kyungyong, for a really excellent seminar. I see all the hand claps coming up from the participants. It was a really interesting talk. Just to remind everybody out there that if you would like to ask questions about this work, you should type into the Q&A box. And I see some people already have. I'm going to, so Catherine Jones and I will take turns reading the questions. Of course, type them in at any time and we'll read them out. So our first question is from Thomas Waxman, who asks, has there been any repetition of this study using the latest tools? And he, since you talked about AlphaFold, he's asking about language-based models and FoldSeq.
Yeah, AlphaFold was certainly able to capture much larger effector clusters. For instance, for max effector, while TR-zeta predicted about 20 max effectors or so, based on AlphaFold model, the max effector cluster was larger as a 30 or so. So certainly there was something that we were missing when we were using TR-zeta. We haven't tested language-based model yet. And I think this will be a pretty exciting story and study if you happen to use a language model and to see how the performance will differ.
Okay, so the next question is from Adrian Hamangian. Did you observe a correlation between cluster diversity and host spectrum when you compared fungal species? So do fungi with broad host spectrum have more clusters compared to fungi with fewer hosts?
That's a good question. First, what we noticed was that we didn't really identify any clusters where the members, so we didn't really identify the cluster that are specific to the host ranges. So for instances, even though some pathogens may have similar hosts, they don't necessarily have a cluster that are exclusive to these pathogens. So, and what we rather saw was that the composition of secretome of a fungal pathogen is likely to follow the evolutionary distances between the species, meaning the pathogens have diversified what is given to them, rather than convergently evolving, effectors that might be useful to infect a certain host. That was the observation that we made. Other than that, we didn't really think about how we can correlate, the secretome diversity or cluster diversity to host spectrum.
Interesting. I'm also wondering how all of this is going to do when you look at secretome of I noticed you included non-pathogens, and it'll be interesting to see whether when you include symbionts, such as, maybe mycorrhizal fungi or whatever into that, because we know that they're also signaling with the host. I'm really excited to see where that takes you. I don't know if that's something you're thinking of doing, but that's something I'm curious about. Okay, next question from Ankit Kumar asks, from identical sequence homology among different effectors, can we assume that different effectors would have similar pathogenic potential? And I'm wondering whether he means actually identical structural homology.
From identical sequence homology among different effectors, can we assume that different effectors would have similar pathogenic potential? Well, I think the point here is that if divergent evolution drives effector evolution, even for structurally similar effectors, their pathogenic roles would differ. So maybe if there are great sequences similarity, we could say their functions might be also similar. But when we move on to structural similarity, very likely we're going to see the functions are already pretty diverse and diversified.
Yeah, it's very interesting because, of course, it's not just what's happening on the pathogen side, but what's happening in the host, right? So even if the effector structure is similar, it might be interacting with something in a different way in the host. Okay, Kay.
Yeah. So the next question is from Yulin Jia. Great talk. What are predicted evolutionary mechanisms of the effectors, the role in pathogenicity and/or fitness?
What are the predicted evolutionary mechanisms of the susceptible effectors? Not really getting that question.
Okay. I think maybe what might be the role of these conserved structures in pathogenicity and fitness for the fungal pathogen? I think that's the question.
I think that's quite hard to say. So for instance, in case of the TIN2 effector that I introduced in Eustylagomatis, for instance, the authors have reported that TIN2 has a very strong violence functions, but that was not necessarily true for the other sequence unrelated structurally similar effectors in the same clusters. So that indicates that there are varying degree of the virulences, even in the same susceptor group, and that will have different impacts on the pathogenicity and fitness. But I don't know if it can be generalized into other types of susceptor clusters.
Could I ask a follow-up on that? So given that you've found that so many of these things appear to be pseudo-enzymes rather than enzymes with activity based on the structural prediction, do you think that there's a lot of them that will be functioning by titrating out some important host function or binding to host proteins and preventing other interactions? Is that something that you think, given that they look like pseudo as enzymes but don't have enzymatic activity, so are they keeping important host proteins sequestered, or is that a possibility?
I think that's an interesting point. So not everything will be pseudo-enzymes, but of course, RNase-like effectors from powdery mildews or the ARTs from Magnaporthe oryzae could be good examples of pseudo-enzymes. And one of the possibility, of course, is expanding recognition specificity towards host proteins that have also similar structures. Then that's something that we're investigating in the lab right now. But I think what might be driving the pseudo-enzyme evolution in general is that once this pseudo-enzyme has an important target in the host that can lead to increase the fitness, that leads to duplication of many pseudo-enzymes. And then these pseudo-enzymes will try to diversify to recognize other similar host proteins, again, that can improve the fitness.
Okay, thanks.
Okay, I'm going to have a follow-up on your follow-up. Sorry, we will get back to the Q&As now, but such an interesting set of ideas here, Kyungyong. So I was thinking your TIN2 example reminded me of a study that Regina Kammen's lab published not long ago, maybe a couple of years ago, where didn't they reconstruct the ancestral TIN2, like an ancestral, it was in Ustalago, I'm going to get this wrong, but it was like an ancestral gene that led to the difference between the way Ustalago affects reproductive tissues and some other fungus doesn't. Have you ever thought of trying to apply that, sort of going back to predicted, that might be kind of fun evolutionarily, going backwards to try to apply your structural models to predicted sort of last common ancestors? I wonder if that would help figure out what changes.
And figure out what phenotypic changes that could have resulted in? That's an interesting question. No, we haven't really thought about going up on the evolution. I think one big reason is that we don't have a lot of phenotypic data. So even though we were able to identify how this evolution happened, we don't really see how virulence functions have been changing and how that have affected the phenotypic results of the pathogens. So we haven't really thought about that at all.
Yeah, there probably aren't really enough to do that with, but that one could be fun to try since you already have TIN2 beautifully modeled. Okay, time to move on. We have another question. Nice presentation. Could you please summarize the overall workflow? So like from source to sequence to the end, and was there any wet lab work involved?
The workflow itself is actually very simple. We first start by collecting the proteomes and then extract the secretory proteins, let AlphaFold to predict them. And then we will compare the sequence of similarity based on some different methods. 1 blastopy, 2 HH blitz, and 3 HH search. We do this sequentially. And this is because each sequence-based similarity search algorithms perform differently to detect the sequence of similarity at a different degree. So for instance, the blastopy search will be the least sensitive sequence of similarity search method, but by moving towards HH blitz, where we compare sequence to sequence profile, and to HH search, where we compare the sequence profile to sequence profile, we can recover deeper evolutionary relationship. And on top of that, we overlay the structural similarity search results that we obtained through TMO line. And then we do clustering using these algorithms. And in terms of experimental side, we haven't presented anything, but we actually have something going on in the lab to test the interactions between the suscept vectors that we're interested in and also the plant targets that those suscept vectors might be targeting. And we're actually seeing some positive results that the suscept vectors of interest are interacting with the plant proteins.
Wonderful. So this was everything you did is a computational approach, but it led to predictions that you're now testing. So I think that sort of sets how it goes. Okay.
So there's a question from Ruben Betts. And did you look into the folding of effectors together with their putative interaction partners, as in making heterodimers? So I think that's a really interesting question.
Yeah, that's actually a really good question. And there was actually the step that we needed to pursue after this study. So for the positive and negative data sets we have collected in the interaction of suscept vectors and plant protein targets, we're following up to model them with AlphaFold multimer to see whether the prediction can actually capture these models. Other than that, we haven't tried anything, but what I can tell you here is that we've tried modeling the protein complex between the suscept vectors and their cognate receptors when they are known. However, AlphaFold was not able to really predict the correct fold for NLR and their vectors. So for those, at least I know that AlphaFold is not going to really work well to model the heterodimers. And I think that's because suscept vectors don't really have a lot of homologous sequences from which we can extract evolutionary information. And then the evolution that is happening between the protein targets and suscept vectors are typically crazy enough to kind of ruin the signal. So I feel like I have growing doubts on AlphaFold performing well in modeling of the interactors from pathogens and plants. But we will see more data soon.
Excellent, very interesting. Okay, the next question is, what was the overall time period required for the completion of work? And maybe you could break that down into how much time did it take for your the work for the MPMI paper, and then for everything up through what you've presented. And I think since I believe you only started graduate school when? Fall
2020.
Fall 2020,
August.
so it couldn't have been longer than that. Okay, so what was the time period required for the completion of the MPMI paper, the work for it?
I think from start to end, it took 4 months or so. And I spent about 2 to 3 months in the protein structure prediction. And I think this was actually same for our bioarchive paper where we use AlphaFold. Protein structure prediction took quite a lot of time. And what was time consuming was not the protein structure itself, but the collection of homologous sequences from the databases. So that took a lot of time. And that can be actually accelerated through, by incorporating other faster sequence and similarity search tools, or using smaller databases. So I would say, also if you're using language model, it's actually very, very fast. For the proteins that are size of 100 amino acids or so, it takes only about 20 seconds, and you don't even have to collect all the homologous sequences. So that can accelerate your work pretty fast too. And the databases that I've highlighted was AlphaFold, the structured database, and then IS-MPMI metagenomics atlas.
And I will say that this will be posted as a recording that will probably come up in a day or two, and that you'll be able to rewatch all those parts where you want to get exact names of things as well. And that will be helpful.
Okay, so the next question from Lee Jun Ma. Excellent talk and wonderful work. Congratulations. Will the clustering algorithm, be available for the community to search proteins of their interest to your defined structural fold?
So we used MCL as our clustering algorithms, which is already available. And then the code that I used for clustering is also available in Zenodo. So you can see the script and then the pipeline that I built for that. And also the clustering output itself, where we list all the effector structures based on their membership in the sus-effector clusters, that it's also available as data sets in the Zenodo. So I think if you go to our bioarchive paper and then look at the last section for the data availability, you will be able to find a link to the data deposit section.
And so that's in the methods for the bioarchive paper.
Mm-hmm.
Okay, thank you.
Hey, have we covered Anil's question? Structural clustering.
I think we skipped. I think that got skipped. I'm sorry.
Pardon me. That was probably my fault. So Anil Girija asks, how can structural clustering give any insight on evolutionary relationships?
So as I said, if effectors are diverging rapidly, leading to loss of sequence and similarity, the only way to recover that evolutionary relationship is through structures, right? So by comparing structural similarity and then identifying which effectors are related by their structures and not primary sequences, we're actually recovering deeper relationship, a deeper evolutionary relationship of these effectors.
Could I ask a follow-up to that?
Mm-hmm.
How will you distinguish convergence from divergence? Is it from, anchoring sections of the protein where some little invariant region is detectable, and what's the best way to divide proteins up in that way?
Yeah, that's actually really a good question. There is currently no clear way to distinguish convergent evolution and divergent evolution. When we talk about divergent evolution in our paper, we typically had to bring some other evolutionary features, for instance, pathogen-specific expansion, or genomic clustering, right? Or you can also consider you can also consider expanding, including a lot of other fungal species with the closer evolutionary relationship, and see genomic context or synteny to provide further support that this is from divergent evolution and not convergent evolution. Because there are now so many structures available, we may be able to actually see what are the folds that unrelated primary sequences may adopt frequently. And those types of information may be able to tell us which effectors are likely coming from convergent evolution, for instances. But I think the repeated occurrence of certain type of such effectors is much more well supported by divergent evolution, because if you think about different effectors emerging in different regions of genomes and independently adopting a similar structure, that is actually evolutionarily more unlikely. So I think there is a little bit of this thinking processes that justify whether this is divergent evolution and convergent evolutions based on the evidence.
Thanks. Okay. Great talk. Within effectors that enter host cells, so cytoplasmic, have you observed any structural similarities that might be related to uptake or translocation rather than function? And that is from Miriam Rafiki. Sorry.
Yeah, that's a good question. We haven't actually looked anything on the localization signals and how those localization signals might be reflected in the structures. So I think that should be a future work. Currently, I don't really have any answers for that.
Really interesting question, though. So many different directions. That's the exciting thing about this work, is there's so many different directions you could go from this stopping point that you have reached already. So lots of cool things. Dave Berger asks, did you pick up clusters of sus effectors that have antimicrobial activity? And that's interesting. That's the whole microbe interaction, one of our top 10 questions.
So some of the effectors, especially the effectors that form pretty large clusters, were annotated as yeast killer toxins. But we currently have no evidence that whether they will be effective against other bacterial species or not. So that might be something that can be done in the future. But I think some of the proteins, some of the secretory proteins may be related to other types of interaction rather than pathogen, host pathogen interactions. So it will be interesting to see what are those effectors are.
So from Diana Gomez de la Cruz, fantastic talk. How do you define the thresholds for clustering, especially for distantly related species? So effectors from Ascomycota versus Basidiomycota fungi that may have evolved similar structures, but due to evolution and host adaptation have acquired certain differences that locate them in different clusters, but they actually have a common origin.
That's a good question. The cutoff that we used for structural similarity searches and clustering is pretty consistent across the fungal species. And indeed, we saw many clusters where the members from Ascomycota and Basidiomycota were clustered together. So I don't think the clustering criteria may be necessarily stringent in clustering of Ascomycota versus Basidiomycota in comparison to clustering, members within the same division. But I think that's actually a good question. It's something to think about.
Lots of good questions. I'm going to ask, we're going to answer a few more questions, but before we do, we'll be posting the link to Kyungyong's MPMI paper in the chat so that you can follow it for more questions. And of course, at that point, you can also find the email for the corresponding author, Ksenia Kresileva, if you have more questions. Lots of good discussion coming out of this. Jane, I'm not sure about this, Chepsergan says, great talk. what is your take on intrinsically disordered regions in effector proteins function? And were you able to capture these intrinsically disordered regions in your prediction?
I think the intrinsically disordered regions, well, we have a few hypotheses. One hypothesis is that, well, based on my experience, I know that there is a set of intrinsically disordered regions that are highly expressed during pathogen infection. So it seems like to me, some of these disordered regions, some of these disordered proteins are utilized for pathogens virulence as well. Those disordered proteins might be sticky, interacting with a lot of different random host components and thereby interfering host cellular activities. In some cases, there are also proteins with ordered, foldable ordered region plus disordered region. In that cases, disordered regions might be utilized for translocations or some sort of protection against the immune systems by interfering the interactions between the core fold and the immune systems. Or maybe it had some other functions. Typically, if you use AlphaFold 2, these intrinsically disordered regions come out with a very low scores, prediction accuracy, because it doesn't really adopt a single foldable structure. At the same time, you might be able to consider some sequence-based predictors, because this predicting disordered regions based on sequence information is also pretty accurate.
So the next question is from Xiaowei Liu. What score system is used to measure protein structural similarity? So what scores can we say the two proteins are structurally similar?
Here we use the TM align, and then TM score greater than 0.5 out of 1 can tell us that they have a fold level structural similarity.
Okay, we are going to answer one more question, but before we do, I wanted to ask Kyungyong if you have, as a grad student award-winning, writing this grad student award-winning paper, do you have any suggestions for grad students? Because I know we have, I'm sure we have several on the Zoom seminar right now.
Suggestions.
Yeah, just like, how did you wrote this great paper, you and your advisor. Clearly you had a great idea, you worked hard, but do you have any suggestions for graduate students writing a paper, their first paper?
Well, first, I think I have to say I was pretty lucky to start my PhD on the right time with the right idea. But at the same time, I spent quite a few years before I started my PhD. I spent 3.5 years in Brian Statsky's lab studying plant immunity on fungal pathogens before I started my program. So I kind of had an already idea what I wanted to do. But I think one of the things that really benefited me was my interest in most recent technology. So like protein structure prediction, for instance, is catching up AlphaFold when it first came out. And then having the ideas and hypotheses to which I can apply the protein structure prediction was really leading me to write these papers pretty quickly and pretty efficiently. So I guess I would want to say, be interested in other stuff than what you are currently doing, and think about how those methodology or tools can be applied to your study and to your favorite questions.
I liked that answer also because it was clear as you read the paper how your hypotheses were so clearly defined and really shown through. And I think a lot of new grad students are interested in learning techniques, but the key is to find the right technique or the right approach to answer the questions you want to answer. And I think that's exactly what you did. And it led you into a very new area. but was, of course, very successful. So I'm going to read the last question from our Q&A. And while I do, I want to also ask people, if you've listened to this talk and enjoyed it and would like to help make what's new in MPMI more accessible, we are looking for someone to help us to edit the transcript. We can send you the transcript. There will be some editing involved. Basically, Zoom does a pretty good job, but it does very badly on scientific terms, which of course a scientific seminar has a lot of. So if you would like to volunteer your time to help us make this more accessible, then the captions, the transcript will make this more accessible to people who are hard of hearing and also people for whom English is not a first language, where it can be easier to read it. Okay, so think about that. If you're interested, please email me, or I think our chat is closed now, so I think you're going to have to just email me with that. Okay, so last question. What language model, it's a very specific one, what language model did you use for structural similarity, the structural similarity search? And is there any algorithm present for structural based clustering?
We didn't use a language model for structural similarity searches. We rather used TM-Align for structural similarity searches. For language model, what I was trying to say was that you might be able to apply language model based prediction for as an alternative of AlphaFold 2, right? And also, currently, I think Fold6 is actually a good structure-based clustering algorithm. It's really fast, and it already has a pre-curated databases that you can search against your proteins. So that might be a good option. Jane, you're muted.
Thanks, Kyungyong. I was starting to reply to that last question, but basically, we realize that these times of our seminars are not always convenient, and there are many factors that go into it, so we apologize for that. But we are, everything will be recorded, as will this one. So I wanted to thank our wonderful presenter, Kyungyong Seong, not only for publishing his very fine work in MPMI, but also for the excellent presentation and really wonderful handling of the questions at the end. And also, I think for your positive attitude, because I think there are not many graduate students who would say, I was so lucky to have started my graduate program in fall 2020, which is something that stuck out to me. Anyway, I have heard, as chair, I have heard many people say the opposite. Anyway, okay, and so that really kudos to Ksenia for obviously having made that a wonderful experience. Kay, do you want to add anything?
I just thought it was very interesting, and I was also thrilled that you said that being interested and looking at what other people are doing and looking for, you know, the right tool at the right time, and that's so important. I was so glad that you recognized that and said that. So
yes, and framed it in that way. Okay, well, thanks everyone. We're going to stop now, and thanks for all the extra applause for Kyungyong for the excellent presentation. You would never have guessed he was a graduate student, new graduate student, would you? Newish, mid-career. Anyway, and we hope that we'll see you on January 17th. If you would like to help out with the transcript, please let me know. Hope to see you all in January. Bye.
Bye-bye.